आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Indian Statistical Institute is a public university which is recognized as an institute of national importance. It grew out of the statistical laboratory set up by Prashanta Chandra Mohananobish in Presidency College, Kolkata. Established in 1931, this unique institution of India is one of the oldest institutions focused on statistics. Primary activities of ISI are research and training of statistics, development of theoretical statistics, and its applications in various natural and social sciences. In this podcast, we welcome Dr. Raghunath Chatterjee, who is a professor and head of the Human Genetics Unit at Indian Statistical Institute, Kolkata. Sir talks about genomics and epigenomics in human health and disease in this episode.
Myself, Raghunath Chatterjee, currently professor and a head of the Human Genetics Unit, as you mentioned. So I did my master's in physics, and then I started working. My PhD was in biological science. Mainly I focused on the genetics, genomics. And after completion of my PhD, I went to USA. I worked at the National Cancer Institute, USA, and I worked mainly on the epigenetics over there and transcription biology and how the gene regulation is actually governed by the epigenetic mechanisms. So after staying over there for around 5 years, so I came back to India and joined Indian Statistical Institute Human Genetics Unit. So and primarily working with genomics and epigenomics in human health and diseases. So currently we are focusing on 2 diseases. One is psoriasis, which is a skin disease, and another is oral cancer. And most of you know the oral cancer, which is one of the most prevalent disease among the men in India. So mainly I'll be talking about primarily these 2 different diseases. So I'll be mainly, first part will be on epigenetic mechanisms in psoriasis, and I'll just try to explain in a layman language that why actually I started working with the epigenetics in psoriasis. So as you know that psoriasis is a skin disease, and in this disease what you see in the skin, skin lesions form. So what happens, so in skin there are primarily 2 layers. One is the dermal layer, and the top layer is called the epidermal layer. And in epidermal layer, in the basal layer, those cells, the keratinocytes, these are called keratinocytes. So these cells hyperproliferate. Whatever we see in comparison to that normal homeostasis of the skin, so there is a hyperproliferation. So because of that hyperproliferation, so the cells are replicating in a faster rate. So what happens? So there is a layer of skins, so they pile up in the top of the skin. So that's the reason we see a scaly silvery plaques in the top of the skin. So these are essentially the phenotype of the disease. And the interesting part of psoriasis is that, so on medication, after treatment, the skin phenotype, it gets reversed. And again, after some time, the patient actually see the reappearance of those phenotypes. So the disease is a chronic disease in nature. It never gets cured. But in some people, we see that this particular phenotype is coming back in maybe 6 months. In some cases, we see after 2 years also. So it's a chronic disease, and the patient has a huge pass through a huge amount of mental stress and trauma, because in the skin surface the scales are there, so then try to use cover the affected regions, and there is a mental stress. So those are the real problem. And other problem is it's a burden for the medical fraternity also, because the dermatologist is so the same patient actually is coming up to the clinic repeatedly because the disease is reappearing. So these are the two issues. But if you think about that this disease, essentially the phenotype is reversible in nature. And as I trained in my postdoc on epigenetics, so the DNA methylation, that was the primary area of research I work over there. So that phenomena is also reversible in nature. So what I thought initially, so I talked with the clinical collaborator, dermatologist, and the idea we came up that let's try to see that as the disease phenotype is reversible in nature, and this DNA methylation, which is epigenetic mechanism, that is also reversible in nature, whether these epigenetic changes are actually regulating the disease phenotype or not. But the question is that how to see the disease phenotype. So the way we actually formulated it, that instead of taking the disease phenotype, so if you take a skin biopsy and see the different histopathological features, so those are essentially the indicator of the disease. So those phenomena we can actually quantify. So we quantified that, and we took the, so initially we did like 24 normal and 24 disease skin biopsy samples, and those did a molecular level testing. So I collaborated with SSKM hospital in dermatology department of the SSKM hospital in Kolkata, and took the samples from the patient with their consent. And then we did a molecular level analysis by studying the whole genome level epigenetic changes, the CPG methylation, and then identified that what are the CPGs which are actually correlated with those different histopathological features, which are also reversible in nature. And we identified very specific CPGs and the corresponding genes, right? So that is what actually regulating the phenotype. So those things we identified, and identified that yes, with the these different the genes which are regulated by the epigenetic mechanisms, they are actually responsible for this disease phenotype. So that was one of the first idea actually we floated, and you can consider it to be a path-breaking work. And we published it in 2018. We published it in Clinical Epigenetics, one of the leading international journal. And after publication of our work, we found that it's one of the highly cited paper. And we found that a lot of people across the world actually they are using the similar concept, because here the most important part was the concept. So that concept was adopted by many other groups, and they also used similar kind of concept to actually work on this particular aspect, the epigenetic mechanism and the reversibility of the histopathological features. So that is one of the achievement. And this part was actually funded by SERB, DST, Government of India, Science and Engineering Research Board. And that project was funded back in 2016. It was until 16 to 19. And I'm happy to share that that project was very successfully conducted. And because of that, actually, in this year, I got Science and Technology Award for Research, which was given by SERB, DST. So that is one of the area of work. And in addition to that, we are also working on the genomics, a lot of other stuff we are working on psoriasis.
Sir explains the scope of translating his research into practical application and the possibility of academia-industry interface.
In terms of the translation part, so should be very frank that we are not there yet, but definitely we started a new concept and the other people also working on this. So what we are hoping that, like in psoriasis, what are the treatment strategies? A lot of biologics are there, but these are very costly, and most of the time, at least in our clinical collaborators places, we have seen that methotrexate, the drug has been given, right? So it's a general immune suppressor, and there is a limit that this much methotrexate in a whole lifetime one can consume. So some restrictions are there over there. And the reason is the treatment is not very specific. Now, what the treatment is doing, the treatment is basically reversing the phenotype, because if you can cure the disease, then it will not reappear. As it is reappearing, that means you are essentially reversing the phenotype of the disease. Now, if we understood, as we understood clearly that these are the different molecular mechanisms through which they are also reversible in nature. So, through which these epigenetic, through these histopathological features and the disease phenotypes are actually getting regulated. So, in near future, we come up with some certain targets. So, if we can specifically target those regions which are also reversible in nature, so that we can actually specifically develop some kind of treatment strategy. But we are not there yet. I should be frank enough to say that we are far, far beyond there, because there are a lot of different kind of molecular level studies need to be conducted, and we are still conducting or working on that area of work.
We are immensely grateful to Professor Chatterjee for his excellent deliberations.